Good day. This is the continuation of the lecture on the cell. The discussion now will be on the organelles found in the cytoplasm. The largest of the cytoplasmic organelle, the endoplasmic reticulum, is usually not visible in histological sections when viewed under the light microscope. What can be visualized will be areas of cytoplasmic basophilia that is due to the presence of ribosomes containing nucleic acid that reacts with the hematoxylin dye. Under the electron microscope, this organelle appears as branching membrane-bounded tubules. In some places, the tubules are widened into flat saccules called cisternae, which may be arranged in parallel arrays. The rough endoplasmic reticulum has its surface studded with ribosomes, while the smooth endoplasmic reticulum have none. The two forms are continuous with one another. The rough endoplasmic reticulum have ribosomes attached to the outer surface of its membranes. It is responsible for the synthesis of proteins for export out of the cell and integral proteins of the plasma membrane. This organelle is abundant in protein-secreting cells. Due to the rich amount of nucleic acid in the ribosomes, the rough endoplasmic reticulum contributes to the cytoplasmic basophilia. Ribosomes are macromolecules composed of ribonucleic acid and proteins. It is not membrane enclosed. It has two distinct subunits. The ribosomes may occur freely in the cytoplasm associated with the messenger RNA. These are called polysomes or polyribosomes, or it may be attached to the rough endoplasmic reticulum. There is absence of ribosomes in the membranes of the smooth endoplasmic reticulum. It is involved in the synthesis of phospholipids for all cell membranes and play a very important role in the liver on the detoxification and metabolism of alcohol and lipid-soluble exogenous drugs. Golgi complex or apparatus is present in nearly all cell types but abundant in cells that produce a lot of secretion. It consists of a stack of 4 to 8 parallel membrane-bounded flat saccules or cisternae. These saccules are slightly curved on one surface and is called the forming or immature phase or the cis Golgi. It receives the protein synthesized by the rough endoplasmic reticulum. The other end of the Golgi is concave in shape and is called the maturing phase or trans Golgi, where the processed protein is released to the cell. Newly synthesized proteins are released from the rough endoplasmic reticulum. The proteins are transported by vesicles as it goes towards the forming phase of the Golgi complex. It fuses with the cisterna on the convex side. It goes down to each succeeding stack for further concentration, modification, sorting, and packaging. The processed proteins exits in vesicles at the concave or mature phase. These are transported to their respective destinations. Some of the proteins from the Golgi complex are destined to be secreted, while others will become components of other organelles such as the lysosome. The Golgi complex does not take up stains very well, but it shows a large, relatively clear perinuclear area against the basophilic cytoplasm of the plasma cell. This is called the negative Golgi image. Mitochondria are organelles that generate the chemical energy needed for most of the biochemical reactions taking place in the cytoplasm. They are present in nearly all cell types but are most numerous in cells that have high energy requirements, such as muscle. It may exhibit variations in shape and it moves around the cytosol. Each mitochondrion has two membranes, a smooth outer membrane and an inner membrane plicated into folds called the cristae mitochondrialis. The cristae has a high content of enzymes for oxidative phosphorylation. Cell types that have high energy needs have a larger number of cristae than those of cells that are less active. There is a narrow intermembrane space between the inner and outer membrane. It has no visible content. The interior of the mitochondrion contains the mitochondrial DNA, which closely resembles DNA of bacteria. It is believed that the mitochondria evolved from symbiotic bacteria that subsequently relinquish the synthesis of their protein in the host cells. Peroxisomes may be present in almost all cell types, but are abundant in the liver and kidney. These are small spherical bodies that contain oxidative enzymes and catalase, responsible for the detoxification of alcohol, uric acid, and toxic drugs. It also breaks down amino acids, uric acid, and fatty acids. The oxidation of fatty acids is an important source of metabolic energy. Hydrogen peroxide may be a byproduct of some of these oxidative reactions, but the enzyme's catalase rapidly breaks down hydrogen peroxide to water and oxygen, preventing its harmful effects. Lysosomes are membrane-bounded organelles usually seen as round dense bodies, and staining properties may vary. It is involved in programmed cell death or apoptosis, which is important in the elimination of damaged cells and for protein recycling. Lysosomal enzymes synthesized in the rough endoplasmic reticulum are transferred to the Golgi complex for further modification and packaging. These secretory products contained in vesicles then bud off from the trans-Golgi. Cell membrane engulfs particles outside the cell and forms an intracellular vesicle around it, called an endosome. The endosome travels through the cell and merges with vesicles containing the enzymes from the Golgi. The vesicle is now called a lysosome when its contents have been digested by the cell. The enzyme starts to break down the engulfed particles. In lysosomal digestion, not all are completely digested. This results in the formation of residual bodies containing indigestible materials. The centrosome contains a pair of centrioles. It is not enclosed by a membrane, and its outer boundary is indistinct. The function of the centrosome in non-dividing cells is to nucleate the polymerization of microtubules that radiate through the cytoplasm. This contributes to the maintenance of cell shape and serves as tracks which transport vesicles and organelles used to move around the cell. The paired centrioles are hollow cylinders with their walls composed of 9 evenly spaced triplets or microtubules. When viewed in cross-section, each triplet is positioned at an angle of 40 degrees from each other. The 3 subunits of each triplet are called microtubules A, B, and C, with microtubule A nearest to the central cavity. Centrioles are important for cell division. Inclusion bodies are diverse intracellular non-living substances that are not bound by a membrane. There are different kinds of cytoplasmic inclusions. This includes stored nutrients, secretory products, and pigment granules. Glycogen is a storage form of glucose which is metabolized by cells to produce ATP, needed for cellular energy. When glucose levels are high, it is taken up and stored by the cells in the form of glycogen. Glycogen granules can only be visualized by using a special stain such as periodic acid Schiff, and shows a magenta-like structure in the cytoplasm of the cells. Triglycerides are stored in the cytoplasm in the form of lipid droplets. Lipids are removed in the preparation of histological specimens, leaving behind only round, clear areas in the cytoplasm if viewed using the light microscope. In electron microscope, they are identified as perfectly round black structures of varying sizes. An example of pigment cytoplasmic inclusion is the lipofuscin pigment or residual bodies. Some cells contain irregularly shaped masses within the cytoplasm that is a result of undigested material. Residual bodies are accumulation of indigestible residues of lysosomal activity and may be found in long-living cells such as the neurons or in alveolar macrophage of the lungs. The cytoplasm of cells contain 3 types of filaments: microfilament, intermediate filament, and microtubules, which constitute the cytoskeleton of the cell. It has several functions, from determining cell shape to movement of organelles and special structures such as cilia and flagella. It is dynamic, with its components continuously depolymerizing and repolymerizing depending upon the needs of the cell. This image shows two components of the cytoskeleton. Microfilaments are the red structures and microtubules are the green structures. Microfilaments are composed of polymers of the protein actin abundant in cells. Actin-binding proteins link actin filaments in different arrangements depending upon function, such as internal support, cell attachment, or cell motility. Short-acting filaments form a dense meshwork immediately beneath the plasmalemma to form the cell cortex needed to maintain changes in cell shape and cell movement. Parallel bundles of actin, called stress fibers, may be found in the interior of the cell that may extend in the periphery of cells. This is important for cellular contractility, providing force needed for cell adhesion and migration. Myosin and actin in muscle interact, resulting to contraction. Microtubules are straight hollow tubules 25 nanometer in diameter with a wall of 9 nanometer thick. They are polymers of the circularly arranged protein tubulin. Microtubule formation is usually initiated at the centrosome. The lengthening microtubules radiate outward from the centrosome towards the cell surface. They undergo cycles of assembly and disassembly depending on need of the cell. It serves as tracks along which transport vesicles and organelles are moved from one region of the cytoplasm to another. Movement is effected by two motor proteins, kinesin and dynein. Intermediate filaments have a diameter in between microfilaments and microtubule. They have similar appearance and diameter, but composed of proteins of widely differing molecular weight. They provide a stable supporting function of the cytoskeleton. Examples are keratin filaments found in keratinocytes of skin, which occupy a large fraction of the total volume of the cytoplasm and protects the skin from mechanical stress. Neurofilaments are another example of intermediate filaments that provide internal support to the axon, which can be a meter long in cells of the spinal cord. As a summary, I have completed the lecture on the histology of the cell. The topics discussed on this second video will be the description and function of the different organelles found in the cytoplasm, the different types of cytoplasmic inclusions, and the cytoskeleton. Thank you.